shortly after we invented it, and as I was gathering my consciousness of the deep diversity of synapses, I realized this was the perfect tool to go to work on that. So hence the overall structure of this. And then this gets into the more computational side of things. So array tomography is a technique that, amongst other things, allows us to resolve single synapses. Here they are in sort of random rainbow colors on this title screen. And this is actually a large area. So we can acquire very high dimensional data about synapses, and we're still scratching our heads over how to do that. And I've heard about 10 really exciting ways I hadn't thought about in my 5 meetings this morning. Then this is kind of a future direction, but one I'm pretty excited about. Discovering biomarkers of synaptic activity and short-term memory storage that will allow us to take yet another crack at this problem of finding the engrams of experience in the brain. That's just a little, coming attractions. But then, as you probably already noticed, I do tend to go on. But I will want help. I want somebody to tell me when I'm within, let's say, 6 minutes of the end of time here. And I will abruptly stop because I want to share with you the new movie, Synaptesthesia. Another word I'm very proud of. This is actually a word that I personally made up with no help from anybody else. And it's a little hard to pronounce, but I think it's really beautiful. And it's based on classical roots, synapse and aesthesia, a Greek word meaning consciousness. And it is a couple of different things, but mainly it is the state that is induced in the viewer by watching this movie. So we're going to finish up with this movie. And the movie will illustrate the use of some of the techniques. And I'll just stick my neck out. I think this movie is actually the most cogent statement that science can make today about the nature of the machine, namely our brains, that creates the human experience. There's one developing very rapidly, forming lots of new synapses. Okay, so we got to talk about synapses. I'm going to start with sort of the old school view. Back in the 50s, the electron microscope and techniques for preparing biological specimens reached a state of advancement in which it was possible to start getting pictures like this. I hope that most of you recognize this as being a close-up picture of a synapse. And this came along at about the same time as Bernard Katz's brilliant experiments discovering individual quanta of neurotransmission, and it was pretty obvious at the get-go that these little circular packets, which you should think of as being spheres, were just the right thing to account for the physiology of miniature potentials. And so this picture is, as I say here, the canonical CNS synapse. And here's just sort of the standard terminology. This would be the presynaptic side that releases the vesicles, and different components labeled as you see here. So there's a pre-side and a post-side. The pulse comes on. That picture is the basis for this cartoon, which is probably even more familiar to all of you. So this is the cartoon version of the same thing, but it's seen in all of the neuroscience textbooks, usually in the first chapter. So we've got the vesicles containing their neurotransmitters, and there's this little gap, the synaptic cleft. And then on the other side, there's some kind of receptors that react with the neurotransmitter molecule and make something happen in the postsynaptic cell. So typically it would work something like this, according to all the textbooks. And I'm not saying this is wrong as far as it goes either. This presynaptic action potential that causes one or maybe more vesicles to nudge around, fuse with the synaptic membrane. We have some outstanding experts in the audience here, especially in the first row, that have studied this process in beautiful molecular detail, causing the transmitter contents to squirt out. They react with the neurotransmitter receptors, and you get a postsynaptic potential. So that's the canonical idea of the synapse. This was sort of, this is present in, for instance, the majority of the computational models of neural network function. It's interesting, but sort of a binary relaying of a signal, the way we have tended to think about it. Okay, back for a second to the real synapse. And I mentioned that a lot had happened in the last 10 years or so. Molecular biology, exquisite tools for finding gene products in and around synapses. And with the help of that, it turns out that, well, there's a lot of really interesting and complex machinery associated with that picture. Here's one of those synaptic vesicles blown up, a beautiful cell paper about 5 or 6 years ago, attempting to render in anatomically correct proportions all the different proteins that are associated with individual synaptic vesicles. Over 100 species, 400 proteins per vesicle. So that pretty hairy-looking molecular machine goes with one of those vesicles, and they all have something to do with its function and maintaining its form. This little fuzz down here by the synaptic membrane is responsible for that coupling of the impulse to the release of the vesicles. And it turns out, nobody has made quite such a nice anatomically correct picture of it. But here's each of those little squares has the name of a protein in it, and people have studied these signaling networks. And suffice it to say, it's at least as complicated. So these are all really interesting protein molecules. They're all switches. They all communicate. They all signal. They tend to talk to one another. I mean, we're a long way from knowing exactly what they're saying to each other, but we know they're the kind of things that communicate in various ways, and we studied piecemeal, and we're still scratching our heads a little bit about how they all interact. But the fact is, they're there, and it's hard to believe that it's an accident that they're all there. The synaptic cleft itself is another whopper thing. In the cartoon, it was just an empty space. As you can see in electron micrograph, there's some density there. There's some stuff there. And in the very best electron micrographs, you can see that stuff tends to take the form of teeny tiny little filaments that line up across to sort of bridge the two halves of the synaptic cleft together and actually maintain their very uniform spacing and their rigidly parallel arrangement. And the fact is, the molecular biologists have told us that there's an extreme diversity of adhesion molecules. I mean, there's cell-cell adhesion molecules in many different families. Some immunoglobulin superfamily proteins, others not. In aggregate, in the CNS, hundreds, if not thousands. There's all kinds of alternate splice variants. And as a matter of fact, there's people, I'm sure there's some in this room, who study the development of the nervous system. That is, the original creation of this incredibly elaborate machine inside our brain cases. And surely there's a lot of variability from brain to brain, but the more striking thing is the similarity from brain to brain. That's why we can all talk to each other. And so the best thought amongst the brain developmental neuroscientists is that the way that machine is built, the blueprint is somehow or other in our genomes, it's in our DNA. It is played out by elaborately orchestrated differential transcription programs in some vast number of different kinds of cells in the brain to express particular subsets of these highly diverse molecules on their surfaces, and that those molecules interact, they guide growing axons and dendrites to the right place, and they mediate the selective partnering of presynaptic and postsynaptic neurons. And so most of these are molecules that we met in the context of studying development. But it turns out that vast numbers, if not all of them, are present in mature brains, at mature synapses, and this is where they tend to be really localized. So we'll come back to that idea later, but the point is it's anything but an empty space. And then the postsynaptic density is the real mother lode of signaling machinery. And here, we studied interactions between multiple kinds of receptors, trafficking receptors in and out of the membrane, all kinds of stuff. And this could be a whole 1-hour lecture in and of itself. But the point is, there's a lot of stuff in the synapse. In aggregate, certainly hundreds in each and every mammalian CNS synapse. We don't actually know that number because we haven't had tools for enumerating them in individual synapses. We enumerate them in biochemical preparations that mix heterogeneous populations. And in in a range of CNS synapses, I estimate, based on careful perusing of the literature, between 10 to the 4th and 10 to the 5th aggregate signaling molecules per synapse. So I would say it's time to forget about the idea of a synapse as a switch, a synapse as a relay, a synapse as a transistor, even though that stood us in good stead back in the 40s, McCulloch and Pitts and so on. The individual synapse is a small microprocessor, a little molecular microprocessor that has all of these molecules going at it in who knows which ways and integrating not just the action potential, but all kinds of signals. And this is the machinery that takes care of that. So, that's 10 other lectures. We're trying to keep our eye on the ball here. Diversity. So it's one thing for a synapse to be complex. They could still be all the same, which means we could come up with a simple idealization and just repeat it 10 to the 15th times and we'd have a human brain. But the evidence is extremely compelling that that is not the case. That complexity also sets the stage for a huge amount of diversity. Now there's two kinds of diversity. There's one kind I call shallow diversity, and this is the kind that we've known about for a long time. So shallow diversity is based on different classes defined by different neurotransmitters: cholinergic, dopaminergic, GABA, glutamate, serotonin, etc. These are famous. A lot of people will explain that the brain works a certain way because it has just the right amount of dopamine in it. I don't think so. I think you've got to have just the right amount of dopamine. I don't think that exactly explains how the brain works. But anyway, this is where that kind of thinking comes from. So that's shallow diversity. But now, if we look just a little bit below the surface at the molecules, we have our first taste of the scary depths of deep diversity. And by deep, I really just, I think it is deep and philosophically deep and all that, and that connotation. I think this has a lot to do with how humans can do what they do and how we can have such rich experiences, as a matter of fact. It turns out that just about every single one, in fact, every single one of those hundreds of proteins in a synapse has multiple genes and or splice variants. There are multiple isoforms of all of them. And we have good tools for tagging them, in some cases antibodies to proteins. There's just a host of evidence. So every one of those classes of molecules that I zoomed over is a potential source of diversity. And in particular, by deep diversity, if we just take one class like the glutamate synapses, which actually happens to be roughly 90% of your 10 to the 15th synapses, there's still that whole range of diversity left. And if synapses were only diverse in their molecular composition, well, it's still something we ought to care about. But they're not. Even within traditional shallow diversity classes, such as excitatory and inhibitory, there is again a deep functional diversity. So today we have magnificent tools for looking at synaptic transmission, our patch clamps, our optical probes, and so on. Brilliant synaptic physiologists working all over the world, and they have studied the function of kinds of synapses in great detail, and the closer they look, the more they see that they're diverse. And these are just some of the axes, everything about the kinetics, the strength, everything you can think of at the level of function is diverse. In many cases, this leads to interesting fights and quarrels in the literature. People are getting different results because they're looking at slightly different kinds of synapses. It takes some years to figure it out. Yet another reason for studying diversity. This is, I'm not going to dwell on any of these pictures, but this is just a little dossier of the kinds of evidence for diversity. There's, diversity of function, as I said, regional restriction of transcription programs in neurons, subcellular restriction of proteins. Here's a case where two synapses onto one cell have different proteins on them, and in this case, regional restriction of proteins in different parts of the brain. So, let's say in this one, in every case, well, for sure, we are looking at glutamatergic receptors. So one of the old shallow classes. But so you can see different alternate gene products are found in spatially patterned ways in different parts of, in this case, a mouse brain. So the diversity is surely enormous. The evidence for it is overwhelming. But we really don't know too awful much about it. And that's the sad thing. It leads to a lot of unnecessary fights and a lot of things just left to misery. And I would say the main reason we remain ignorant of it is there has been a real shortage of tools for looking at synapses one at a time. Most of the, we learned about the presence of those molecules by pooling synapses across regions of the brain and so on, and that tells us that they're diverse, but it doesn't tell us how diverse. You really need tools that will resolve, measure, weigh, etc., individual synapses, and those have been few and far between. There's one that goes back to the 19th century, and even though it is kind of technologically crude, it's still important. There's companies that do this and nothing but this. And this is counting dendritic spines in Golgi-stained preparations. And I think the thing that makes it a good deal is, up until pretty recently, that was the only way you could see individual synapses one at a time. And in fact, it was by staring at this thing that Ramon y Cajal in the 1880s got what we consider today still to be the correct idea of what the brain is. It's a network of lots and lots of individual neurons interconnected at synapses. And he got that idea by seeing these little dendritic spines, which it turned out are the sites of individual synapses, only the postsynaptic part, the way this thing works. Then in the 1950s, the electron microscope that we already came along, and it gives us a dandy view of individual synapses. But up until very recently, its throughput, its field of view has been very limited. And so, you work all day and you see maybe 12 synapses. And you see diversity, but on the other hand, you can't measure molecules too well with that. So that's really important, but not everything we could dream of. Optical physiology, here's a crude early example. Electrophysiology actually isn't so good at looking for single synapses. Ask me about it later. I'm going to run out of time. I'll talk about it now, but I'll throw that challenge out there. There's very little electrophysiological observation of single synapse function. Optical tools, take that as an icon, do give you a better shot at looking at certain aspects of function in individual synapses. So let's see how my time is coming. When is this supposed to be over?
About 3, but you started later.
Yeah, okay. So this gets kind of exotic. I think it's important. I hope you're staring at it. These are just some hypotheses. Once you have that deep diversity in mind, these are some ideas to think about. I may have already spilled the beans on a couple of them. Those cell-cell adhesion molecules and signaling proteins are definitely detectable at mature synapses. They are known as developmental guidance molecules. To say that the molecules detected in mature synapse are the one that built that synapse or that part of the network in the first place, that's a bit of a leap. I think it's a really attractive hypothesis. You can think of other hypotheses. This one has the most appeal, that they simply stick around and are maybe repurposed. Incidentally, most of those things, when they're done guiding development, can be shown to have actual signaling roles in the mature synapse. So that's just one of the tangents flowing out of this. Maybe it's not my favorite one, but it's one tangent about proteomic diversity. This is one I like quite a bit, and maybe this is where the synaptome meets the connectome the most. Those diverse proteins you can find at synapses. Well, we have a biological principle here. We are pretty sure that the way a synapse is made is by two neurons coming together, what we call the presynaptic and the postsynaptic neuron. And we're pretty sure that the proteins that are present in that synapse all came from one or the other of those two cells. And in fact, the ones on the presynaptic side from the presynaptic cell, postsynaptic, and then the adhesion ones meet in the middle. So that's a principle of cell biology. And there are a few examples of proteins leaping from one cell to a completely different cell and becoming incorporated in it, but they're few and far between. So there's all those diverse proteins, and we're pretty sure that whatever you see at one synapse came from either of the, one or the other of the two parent neurons, the mother and the father, or the presynaptic and the postsynaptic, whatever you want to call them. And they were diverse. Maybe they were diverse enough to build the brain in the first place. We know they're very diverse. And so here's an interesting idea, that they are a connectomic code. That you look at a synapse, and if you have the right tool, and I'm going to tell you about one in a second, that can read that code. Maybe it's a code, it's encoded in the presence of a couple dozen out of a potential range of a few hundred molecules. That's a very big code. And that code tells you who the presynaptic neuron is and who the postsynaptic neuron is, or at least what their cell types are. Although we need the proviso that the concept of neuronal cell type itself is still a work in progress these days. But in other words, in every synapse, maybe there's a little molecular barcode that tells you gives you a whole lot of circuit contextual information, connectomic information. I like that one a lot. We don't have the lexicon to interpret it yet, but I've got some ideas about how to find it and some of the things we're working on. Okay, also some of that protein, we know that there's all these things called plasticity, adaptation. We know that activity affects synapses. It changes the way they work. And the prevailing idea about how we store memories, at least short term, is by changes in protein molecules at synapses. Maybe moving a few of them in and out of the membrane, maybe phosphorylating some of them, changes in proteins that potentially could be read in a snapshot. So you look at a neural circuit, and by looking in the right way at the individual proteins, because they are so diverse at individual synapses, you can tell whether that cell was active in the 5 minutes before you sacked the animal, whether it had, stored a memory lately. And that can open the door, combined with, if you had great neuroanatomical precision, which I'm going to tell you about in just a second, some real interesting new opportunities for decoding the organization of neural circuits. And last but not least, we know that synapses can turn over, that new synapses can be formed in adults, that old synapses can be broken, and those events of synapse formation and elimination don't just happen. There's a reason for them. And based on everything I think I know about biology, the reasons are molecular. And it just may be possible to read something about the status of a protein vis-a-vis these sort of protein turnover rewiring changes, the kinds of changes that maybe are necessary to encode long-term memory, the lifelong memories, the memories, as Sebastian Son would say, of your first kiss, the ones you never forget. Maybe those are really rewiring, but synapses are fundamental agents of rewiring. That's the story in development. Connections are made because a synapse is formed and stabilized. Many synapses are formed and not stabilized, and so this connection isn't made. That's how we believe development works. And as a matter of faith, there's got to be a molecular reason for it. Okay, so that's the big, long-winded, longer than I said, manifesto on the subject of synapse diversity. Pardon me, but I just, personally, I feel it's incredibly important. I've got to say it. Okay, now what can we do about it? If there was nothing we could do about it, it wasn't worth saying. I think it's worth saying because we have now at least one, there's probably more, and surely better ones will come along, one technique for sifting through and starting to measure that diversity. And so the one that I know about is called Array Tomography. So that's what I'm going to talk about next. Here's a busy little animated slide that shows all the steps of Array Tomography. I'll let you look at it. What it is a mashup of techniques that were developed and perfected over the last 50 years by electron microscopy, electron microscopists, and a nice dose of modern optical microscopy. And the whole thing works because it is glued together by a bunch of modern computation. The computation is pretty challenging today. The basic idea of array tomography was there in the work of the 19th century histologists, but nothing like this would have been possible without, like, say, the last 10 years of Moore's Law. That's probably why this technique waited for us to discover. Okay, so take your piece of tissue. Now this is strictly a histology. So the first thing you do is you gently and respectfully, as painlessly as possible, sack your mouse or whatever. And then you take out pieces of tissue, you fix them and embed them in plastic the way electron microscopists do. And they've gotten pretty good over the years at developing techniques that just kind of take a snapshot of the tissue and take the water out, replace it with plastic, but leave all the proteins and other biomolecules right where they were so you can study them. And because they're in plastic, you can use a really, really sharp knife and cut them into really, really thin sections, which lets you put them in electron microscopes or do the rest of what I'm going to tell you about. And so we use a version of standard electron microscopic serial sectioning where you kind of soup it up by putting some glue on your block a special way, which causes arbitrarily long ribbons, continuous ribbons of serial sections to form and float out on the water in the little water trough that is one of the fundamental tools of thin section electron microscopy. And then we figured out a way of bonding ribbons of those sections, something like 50 to 100 sections at a pop, very tightly to a precision optical glass cover slip. So they're stabilized and held in place really nicely, and the thing they're held in place on the precision optical glass cover slip is kind of the one place, the one thing where you can get really diffraction limited optics at high numerical aperture. The only way that we'll really do that. You hear diffraction limited imaging tossed around rather lightly, but it's usually aberration. Okay, then you can put that slide in a special little holder and stain it with, well, whatever you want. But what we normally use is antibodies. Oh yeah, sometimes we start off with a GFP transgenic mouse. And we use procedures that preserve the GFP fluorescence through the fixation, embedding, and sectioning. But you can stain it with, let's say, 4 colors, standard indirect immunofluorescence, and then put the array on a microscope. And you get tired of doing this real fast unless it's an automated, motorized, computer-controlled microscope. But those are pretty easy to come by these days. So here's the microscope in real time. You can see it's flashing through 4 colors. It's taking its little field, snapping it onto a digital camera in each of the 4 colors, and then it moves its little automated stage a little bit. And it can tile your specimen, your complete ribbon, or subsets of your ribbon, however you like. And then I'm not going to talk about this much, but it turns out also you can take those same specimens, and if you process the tissue just so, and it's tricky, but you can get it to work. And once again, there's great experts in the front row here. You can take that same specimen and pop it into a field emission scanning EM and get really nice EM pictures. But I'm going to stick to my fluorescence here. And then you get all these little pictures, but now this is where the second place. First there was computer automation here, that's not so hard. But now you use computers to stitch these images back together into their serial sections, their complete sections, and to align them in 3D. So after all this work, you get a multicolor image stack, much like you could get by just pushing one button on the confocal microscope. Now, there's one other special thing. I have to advance the slide. Pay careful attention to these purple arrows. Okay, so this is completely serendipitous, but we discovered that when you have thin sections in this particular resin stabilized in glass in that particular way, it works to strip off your first 4 antibodies. You do that by just changing the pH by a couple log units either way, and you disrupt the fit between the antigen and the antibody. The primaries and the secondaries come off, you wash them down the drain. If you put them back on again, look at it again, you get the same result. But that's not actually what we normally do. What we normally do is we put 4 other antibodies on. And you can repeat this 10 or 15 times. So this method can give you, potentially 40 or 50 color immunofluorescence and more. It's a question of how tight the adhesion to the substrate is. I'll show you some examples of 18 color array tomography in just a second. Okay, so why go to all this trouble instead of using a confocal microscope? Well, I just said one thing. You can get a much larger number of colors. If you're trying to study something like a synapse that has hundreds of different proteins in it, that might be good. But it turns out you get very, very much higher resolution. Partly because when you have to focus down into a 3-dimensional specimen, you get all kinds of optical aberrations, which it's really impossible to eliminate, that cause a very rapid deterioration of the resolution. And basically, there's no way to have your 1.45 NA in a wet specimen at all. And So these are little volumes that were collected of very similar specimens. In both cases, they were little pieces of mouse cortex, and the white spots you see there are individual synapses. So this is one of our favorite nearly universal synaptic markers, an antibody to synapsin 1. So this illustrates, well, for one thing, you can see many more smaller synapses on the left. This is the right answer. They're distributed at relatively uniform density through the thickness of these volumes. And so in the case of the confocal, the brightness falls off for a host of reasons. The antibodies have trouble penetrating through the thick tissue, and the light has trouble getting in and out, and aberrations and light scattering effects reduce the optical sensitivity. And of course, the fact that you eliminate that depth dependence means that it's possible to interpret an image like the one on the left much more quantitatively than you can interpret the one on the right. That depth dependence would be enough of a problem. You can actually see that the depth dependence varies from spot to spot. I think the antibodies find little fissures within the tissue to go deeper in some places than others. So this would be, it's really a nightmare to quantitate. Oops, well, anyway, enough said about that. So this is, let's see, I'm wondering if we could turn the lights down about now. Yeah, thank you. Okay, so this is the native YFP fluorescence of a certain transgenic mouse. This is the work, of course, Thy1, Line H, YFP mouse, looking fun, dated back in Jeff and Josh Singh's lab. And it's just another example of this technique, and you can see that there's lots of detail. It's one of the nicer optical pictures you've seen in the Quacher. We'll see a lot more of this later. Here in the same volume, the same rendering, this is a stain for acetylated tubulin. So the microtubule core bundles of axons and dendrites are acetylated. It's probably the reason they're so stable, or maybe they're acetylated because they're stable. Nobody really knows for sure. But what you're seeing here is, in the previous picture, you saw the green from the 4% or so of neurons. That was still a sparse representation. So that's about 4% of the neurons in this mouse. So this is about the density of the artist's impressions of the inside of the brain you usually see. This is leaving out completely the other 96% of the neurons. Those empty spaces are filled with neurons that are invisible here because they aren't expressing YFP in this particular mouse. They are not at all empty. Everything else is jammed full. Here in that same volume, now we can see the big axons and dendrites of 100% of the neurons. The contrast is nothing like with the YFP, but you can get a sense of how many more dendrites there are running through there. There's these little voids are the cell bodies which don't have acetylated tubulin in them, or some of them are blood capillaries. If I get a chance to kind of, we're still kind of.
Can you turn the lights off?
Can we turn the lights farther down? My projector more. Anyway, this is the synapses and only the synapses. So this particular marker is a phosphosynapsin 1, and it nicely stains at least 99% of the neurons in the cortex, if not 100% of them.
Side ones won't go off.
Maybe it's a fire marshal thing. Okay, well, anyway, a little quick tour through what a piece of cortex was. Well, the point of those synapses, which it was kind of hard to see with that illumination, it's a little easier to see now, is that the synapses, even though they're packed in there at a density slightly in excess of 1 per cubic micron, are individually resolved in 3D. And you can see it especially in the rocks. There's individual little puncta, which are easy to segment out and analyze. Here's one little case of, this is like 9-color immunostaining. 3 panels, the same volume, with different synaptic markers displayed in different colors, because most of this, we have at most 3 different color receptor channels in our retinas. Some of it's only 2. So there's a bunch of interesting synaptic protein markers, some of which tap into that diversity. I talked about it sexually later on. Here's a way that we use in the lab for looking at 18-channel data with nearly 3 colored channels in an eye. So these are little sets of serial sections. There's 2 sets of 9 serial sections here. One centered on what the markers say is a glutamatergic synapse, one on the GABAergic synapse. And the idea is it's the same section in each little cell within the column, and the sections go from -4 to +4 in the rows. And here's a bunch of synaptic markers over here. We'll dwell on them just now. These are the glutamatergic markers that make us think this one's glutamatergic, and the ones that make us think that one's GABAergic. Well, this is an electron micrograph made by scanning EM. This was actually our very first one. We got quite a bit better at this. Here's an attempt to combine immunofluorescence and EM. But as I said before, I'm not going to talk about that today. Okay, so single synapse analysis. Okay, I'm going to go through this real fast. I may completely skip over memory biomarker. So here's a few of those channels, a few out of the 18 selected at random. The chaos of all of those markers in a piece of cortex. One little GFP dendrite, this is that same transgenic mouse. Here's a little bit of a blood vessel, and these are the cell bodies of non-expressing neurons. And these are puncta, couple markers of all synapses, and the blue one is one that only lights up GABA synapses in this case. There's actually many more markers. So what we want to do is to be able to quantify and classify, first a supervised classification based on our sort of naive textbook and Nature reviews ideas about what kinds of synapses there are in the brain. So the approach we used here was a machine learning approach based on supervising by hand classifying a training set. So these little squares were placed. This is actually a volume image in 3D. We're just looking at a projection through it. But there's some randomly placed volumes that we captured and used as a training set to train a machine learning classifier, a support vector machine in the case I'm going to show you. And so here's some more of those synaptograms, like the 18 colored ones I showed you here, boiled down a little bit. Again, serial sections, and it takes a while of staring to learn to interpret these things, but after a while, if you're working in my lab, you get good at it, or die. And so here's one of those things. We have this little computer game. It's lots of fun, and students and postdocs have to play it on their lunch hour. And so they get a deck of these synaptograms, and there's some buttons like glutamate, GABA, or not, something like that. And so the student would look at this one, and they would notice that it's got B-glut1, well, that's a vesicular glutamate transporter. GluR2, that's a postsynaptic glutamate receptor. PSD95, a postsynaptic. Yeah, good enough for me. Okay, so that one is a glutamatergic synapse. That's what makes it so much fun. You get some random little graphic thing happening on the screen. Okay, then next one up, and well, this one's got VGAT and GAD, starting to look like a GABA synapse, and yep, it's got little Geffen postsynaptically, so you push the GABA button, and you can do a couple hundred of those during your lunch hour once you get good at it. And so here's the results of that. Projected, this is the hand classification projected back into the original image coordinates, and now they're displayed as little spheres in different colors, blue for inhibitory, red for excitatory. And so that's all very nice. But now the good part is based on that training set, the support vector machine can define some high-dimensional planes that will, do a passable job of simulating what the graduate student on their lunch hour did. And it won't complain. I don't have to be bribed and coaxed and threatened. It will just go through and classify synapses by the million. So there's a result, and again, red for excitatory, blue for excited, inhibitory. There's some nice things, like it's not trying to tell you there's any synapses inside the cell bodies. that's good. One or two random pieces of junk. And it even shows a slight tendency for the blue, the inhibitory synapses, to encrust the cell body, which as far as we know is the right answer. And then the rest of them are interconnecting that dense tangle of axons and dendrites. And then once you've done something like that and gotten tired of looking at it, you can pull out actual numbers. So these are numbers of synapse densities of various kinds of synapses as a function of depth below the peel surface. The cerebral cortex is a beautifully laminated structure, so that is an axis known to be meaningful. And these are nice reproducible curves, and they're, where there's information of this kind in the literature, these are consistent with it, even though they were generated by this machine-trained process. And so, for instance, one might use this to look for abnormalities in synapse distribution as a function of disease condition, or to try, to begin the work of putting the synapses in place in a computational simulation of a neural network. Have to be done a little bit more finely for that. That is an idea. Here's another fun trick with 3D image processing tools. So there's the YFP in a section, then we use just a little seed filling type tool, and we find one dendrite, only the stuff that's connected to it. We dilate it a little bit and use that to clip only synaptic markers out for further analysis that are very close to that dendrite and have their postsynaptic side sunk into the YFP. So those colorful little spots, a few markers, are the synapses that almost surely are the synapses onto that dendrite. And the point of that, this goes back to that thing I said about inheriting synapse identity and diversity from the parent neurons. This is part of our program to test that idea. And that, in turn, allows you to pull out numbers. These are similar to the ones I showed you before. But now they're restricted to the subsets of synapses that are on to yellow dendrites, which tend to be the layer 5B projection neurons in this particular mouse. And it taken a stab, only a shot so far, at unsupervised clustering or classification approaches. This actually will get much more interesting. So now we've got all this data, millions and tens and hundreds of millions of synapses, and this is a work in progress. And I learned today that there's people in this beautiful building in Utah who know a lot more about this kind of thing than I do, but can use the basically the idea here is to let the data speak as to what there are in the way of clusters or classes of synapses. Something we would very much like to know. I think, my first 25 minutes, I probably got most of you care about such things, real depressed about the diversity of synapses. And I think the way out will be to find clusters or classes, to start simplifying things again. But I think you've got to go through the morass of complexity before the simplified part is really going to help you. I mean, I can make it as simple as I want it just by saying it was so, but that would not make it so. Okay, memory biomarker discovery. You're not really time for this, except for this one slide. I love it so much. The picture on the left shows you that it is occasionally possible to infer dynamics from a snapshot, a single snapshot. So you can tell which spires of this particular famous cathedral are currently being worked upon by the presence of cranes. And there's plenty of things that we could think of as little molecular cranes in brains. Here's one of them. It's a meteor gene product called ARC, and we're really fascinated by this kind of thing. We've got candidate plasticity activity markers and finding them that clearly decorate some but not all spines on a given neuron, some but not all synapses. So it may be speaking to us about which synapses were active, which ones had undergone plasticity. This may be the beginnings of a new way to look at engrams of experience. Here's some of that kind of data. Okay, I think I'm just in time. Yep. Beautiful. It's almost like I planned it that way. Okay, synaptesthesia. I already told you about the magnificent name that I thought up. A little bit more about the movie. So we're going to be looking at this volume. Those little snapshots I showed you before when I was introducing array tomography are a little piece down here. So you've seen this before, and you may remember that the green is the YFP, and it's a few percent of the neurons, mainly these layer 5B projection neurons with these big, tall, beautiful dendrites and long axons. The red is synapses, these little microprocessors, but there's 10 to the 15th of in our brain. And the blue is a faint ghost of most of the large microtubule cores of axons and dendrites. This particular volume was acquired by that microscope that I showed you the little movie of. It sat there for 10 hours and snapped 12,000 individual image tiles in different colors and so on. And then they were all stitched together. The entire volume is 1.5 millimeters high. That's enough to go through all 6 layers of the mouse cortex, through the subcortical white matter, and we pop down into the striate. And in most of the synapse sensory, we hit hippocampus there. This one hit striate, which is really interesting anyway. This volume is, it's got about 6 million synapses in it. This volume is about, the total volume is about 1% of the volume of the smallest fMRI voxel I know about. So I've got to say, I'm somebody who really, I'm very enthusiastic about fMRI. It's part of the great tapestry of ways we look at brains. What we have to understand is how much stuff is going on within the individual voxel. And you don't have I have to show you the movie before you have a sense of how really much stuff is in there. But bear in mind that everything you're seeing is, there's room for 100 of them within one fMRI volume. 6 million synapses. That entire volume is still only about 1/50,000th of a mouse brain. And since our brain is about 3,000 times larger than a mouse's brain, it would be like this whole volume that you're going to see is really big and got a lot of stuff, as I said, is 150 millionth of what's in our brain. And I want you to think about that, just in case you're worried about stuff like where free will comes from, and where creativity comes from, and whether a machine could possibly produce creativity or free will. I think this kind of machine, maybe it could. I think it's not outlandish. And here's some numbers to kind of back that up. If you take the numbers I already told you, 10 to the 15th synapse in the human brain, 10 to the 5th switches per synapse, that works out to 10 to the 20th of those switches in a brain. And that's only the synapses. That's not counting the ion channels and other computational elements in the brain. And 10 to the 20th is getting in the same ballpark as the total number of transistors on Earth. All the transistors and all the computers, memory chips, routers, everything. So one human brain has switches about, it's not analogous to a computer. It's analogous to all the computers on Earth. Of course, it's different because it's on all the time. You know, most of the computers on Earth are turned off, and most of the parts of them are turned off. And it's just possible that evolution has come up with clever algorithms. Now, to help you really appreciate this, my daughter, for the grand sum of $50, about a year and a half ago, this is her, composed a musical score, which we ended up submitting. This movie I'm going to show you, Synaptesthesia, didn't have that name yet. It originally saw the light of day, supplemental video #1 to a Neuron paper. And it was the first time anything ever appeared in the Cell Press with the following caption in the supplemental data. It said, supplemental video #1 is best appreciated with your computer hooked up to good speakers turned up loud. And the editors were going to change it, and the referees said, don't you dare. Okay, so that's her, and she's actually basically a kick-ass rocker. She's a sophomore in music at USC in a really wonderful program. This is her band in the studio last summer. That's most of you are much too young to know who I'm talking about. This is John Fogerty's son right here, her lead guitar player. Really wonderful young man. And they were just cutting their first EP as a band. So that's the real Catherine. So she's kick-ass rock and roller, band leader, but she managed to nail, within the total space of 50 minutes, composition and performance. Pardon me for bragging. I mean, she's my daughter, what can I say? But I think nail something that really conveys what kind of a machine we are when you put it together with this little video. And I think the video, you have all the details. I'm going to shut up and let it play. I think I will just completely shut up and let you enjoy Catherine's original score. But I want to leave you with the idea of thinking about, remind you of what I just said. This is 1/150 millionth of your brain. We're seeing only our flight path through it, where it's a fly-through animation, is only sees about a quarter of it. We're looking at, let's say, 3 elements of the transcriptome. There's about 20,000 of them in a mouse brain. So we're looking at 3 of the 20,000 colors that are there mingling, making your brain, making your synapses work. And the green is only about 4% of the neurons. So it's this very, very sparsified set of 1/150 millionth of your brain. So watch this, and then try to worry about where creativity and free will comes from. Okay, is the lights absolutely as dim as they can go now?
So,
Okay. Even those ones in the back are killer.
seems to be a screen.
There it is. That's a step in the right direction. Okay,
Thanks.
well, look hard, everybody. Okay, here we go. I'm done. But I want to say one other thing. There's a little bit of, maybe a little too much stray light in the room today. And this exact movie is available for download on my website. I mean, it's also on YouTube, but don't just look at the YouTube version because that's over compressed. But you go to my website and you have a good broadband connection, you can download either this one or the full HD one, which is, believe me, much better. And if you burn it, for best results, you burn it to a Blu-ray disc if you've got a Blu-ray disc, full HD one. And that Blu-ray disc hardware can really play the hell out of this thing.
We have time for a few questions before the next class comes in. Questions for Stephen? Eric.
So do you have a way to go back for your automated calls? Is there a measure of confidence that the automated call can spit out, and then can you go and look at the low confidence and check confirm by hand?
Yes. Well, we've got a paper that we're just about to send in class computational biology. It's about all of that. And first off, there is concordance between the 3 graduate school students who generated the training sets, and we looked at different training sets. So maybe that's the closest thing we got to a ground truth is the human classifiers. And then the paper actually compares a whole bunch of different sort of standard machine learning algorithms. I think actually the one that wins in the paper, this was a support vector machine. There was a random forest classifier that had better concordance with the human classifiers. So, there are, of course, it's only as good as the training set generation. But that's basically what we've done, is just to compare different.
But that diversity may be in the marginal calls, right? Those may be ones that are synapse and minus, because these are silent synapses. But all of the cool things you talked about the hypothesis for diversity may be, in fact, in the failures.
Yeah, that's right. This only scratched the surface. I mean, the main real synaptic diversity here is V-glut1 versus V-glut2, which are fairly well known at this point ways of sub-classifying glutamatergic receptors. And the results, our results are very consistent. They're strong in layer 4, strong in barrels. They're, basically the thalamocortical projections. But I mean, that's sort of one out of what I think eventually will be hundreds of examples of where a synaptic protein allows you to make a strong inference about the origin of a projection. So people use V-glut2 as thalamocortical, V-glut1, it's probably cortical-cortical, which are the 2 main things you see here.
Question?
Very good question. As you begin your lecture, you talked about synaptomes, connectomes, and you said there was a 3rd tome, or at least there should be, and I didn't hear you say what it was.
Well, I might have misspoken myself. No, I think what I was trying to say, what you probably quoted me correctly, but what I was trying to say is I wasn't sure whether a synaptome was part of a connectome or whether it was yet another ome in its own right. 2 versus 1. I think that's what I meant to say.
So a question for me. So what do you think is your biggest challenge from taking it to the next level? Is it from the image processing? Is it the computation? Where's your biggest bottleneck now?
Well, okay, one frustrating bottleneck is just stitching together and registering the biggest data sets we've got, which are 30 times the volume. We've got the arrays sitting there. We've got a lot of the data acquired, 30 times the volume we have. This was a little slice through 2 whisker barrels. We like the rodent whisker barrels as a beautiful little Rosetta Stone of cortical organization. So we have been collecting data where we get 2 complete whisker barrels, and in the experimental condition, one of those barrels has been stimulated a special way or had the whisker trimmed off. So we're looking for, that's one of the ways we're looking for plasticity markers. And it turns out that this volume, 1/30th, is about the most we can do with high-end desktop computation, with a couple of 8-core Apple Intel machines. It took 2 or 3 days to register those, and there's all these little things that can go wrong. So I mean, it's this is straightforward. It's not completely rocket science, but it does need good algorithms, and it needs enough computation. And we have kind of given up on registering the 2 whisker barrel sets. So we're falling back to doing a sloppy registration, cutting out regions of interest, registering those. That brings it back into high-end desktop territory. So, I think people here, such as Tolga, have already solved that problem. So I certainly don't mean to say it's a trivial problem, because it's been kicking our asses. But that's one kind of problem. And then, in a way, the more open-ended questions are, how to deal with that diversity. And I'm sure the answers there will be computational. They will be, unsupervised clustering and, finding ways to hear what the data is trying to tell us. I can easily sit here right now on the blackboard and make up a scheme for classifying all the synapses in the brain. But nothing is going to work in the end unless it's the right scheme. And I think, we have to figure out how to let these terabytes of data tell us what the right way to classify them is. The data is, a little bit noisy, uncertain, and we don't know where we're going. In terms of that, so I think that classification or clustering problem is the biggie. And what I mentioned, I like using the computational tools to check things out, but I like using cell biological principles where possible too. So the idea that any given synapse inherits its proteins from only 2 neurons, and we can learn something about the neurons, and there's a big saving right there because there's roughly 10,000 synapses per neuron. So if we can learn the rules relating synapse, neuron to synapse, we've made some progress. And then, most people believe that there are, classes of neurons or types of neurons with, large numbers of instances of them. That's, a dream that's been around since the 19th century. It was one of the founding dreams of neuroscience, that there were types originally defined by morphology. But, the trouble there is, as we get more and more powerful molecular tools for, really looking at the phenotypes of neurons, the old classifications are breaking down all over the place. I mean, there's more to it than that. But so, I think that, the really hard problem of, the big problem is managing the complexity of the brain. I mean, in general, that's the problem with the brain. It's good news that it's complex enough to be a viable solution to understanding the human experience, which is what we want to do, and our own subjectivity. I think it's plenty complex for that, but of course, that's a two-edged sword. The other side is it's too complex for us to understand or begin to think about. So number one problem is managing that complexity, and I think, what we need is complexity management principles, clustering, classification. Those are the tried and true solutions that the human race has used from the beginning of time to deal with the complexity around us. But I think this is a lot more complexity than we tried to deal with before. And so I think, that is the real challenge. The little piece that I see right now, I have my eye on, is unsupervised clustering, but it's really complexity management.
All right, well, let's all thank Stephen for that.